Now I'd like to introduce Dr. Craig Garner. Daria just mentioned some of his work was inspired and sparked by the SPARK program that you just heard about. He's a professor, up until recently actually, in the Department of Psychiatry and Behavioral Sciences and the co-director of the Center for Research and Treatment of Down Syndrome at Stanford University. He's recently moved, however, to Berlin. Perhaps he'll tell you more about that when he speaks. He is a developmental neurobiologist. He's been interested for a long time in how environmental and genetic insults to the developing nervous system impair cognitive function, including in disorders like Down syndrome. And I'll welcome Dr. Garner to the stage. Thank you for being here.
Thank you, Dennis. Good afternoon. I hope you're having a great day. As parents, we have great expectations for our children. We expect them to be born healthy. We expect them to grow strong. We expect them to have friends, to go to school, to do well, to go and learn how to play the piano or the guitar. We expect them, hopefully, to go to college where they'll meet the love of their life and get married, have children, and have a very happy and successful life. This idea is one that is shared also by children, our families, and children with neurodevelopmental disorders, including Down syndrome. They have dreams that these children will be born healthy, that they will be able to interact with their grandparents, that they will have friends that they can associate with and interact with, and that they will also be able to go to school, maybe go to college even, and ultimately to have life fulfillment by a job or friends or marriage. This reality is, however, far from the truth for this population. There are exceptions for this population, but in general, there's an overall skepticism about what one can be done. What I'd like to talk to you today about is that this concept is changing within this community. It's being driven by the parents, and it's been driven by the scientists and clinicians who are now slowly recognizing that while complicated, and I'll tell you a little bit more about that there is like true hope for what you can do for even such a complex disorder as Down syndrome. So let me give you a little bit of background. You probably all know Down syndrome, probably have friends or family with children with Down syndrome. It's very complex. It involves the triplication of an entire chromosome. And this chromosome encodes over 300 different genes. There are many systems affected by this triplication. Some of them include bone development, which affects their craniofacial features. It affects sleep apnea. It affects heart development. And for this topic, it affects brain development. In terms of brain development, the average IQ is somewhere between 70 and 60, but can be more severely impaired. They have other associated disorders, including autism type of spectrum disorders and ADHD-like behaviors. They have a primary deficit in speech and language, which is rooted in the fact that they have very poor learning capacity. Their short-term and long-term learning potential is very, very small. You try to teach them cards, you try to teach them names, teach them things, they can't hold on to this information. Further bad in this particular scenario is that by 35 to 40 years of age, they will all have a pathology of Alzheimer's disease. The current standard of care for this population, which actually only really got to be better in the late 70s, where before that most of these children were institutionalized, is through better nutrition. They're being kept home. They're actually through inclusion education, are being placed in enriched environments, and you heard how important enriched environments are. This has been one of the game changers for this population. Many of these children are doing much, much better and are actually going to school and having jobs. So the question then is can we do more? And what I'm going to tell you a little bit about is what I call the translational cycle, which I got involved in because we were doing some basic research to try to understand what was the underlying cause of why the cognition was reduced and whether we could do something about that in a pharmacological way now, independent of their age. We don't have to start as babies, could we start older? And it requires that we actually understand more about what's wrong with the human condition, to understand using animal models, to define mechanisms, and hopefully to develop rational therapies. So as I mentioned already, there are many genes involved, and we need to know what these genes are and what they're encoding for and which ones are important for cognition. The brain is affected. We have a smaller brain in the case of Down syndrome. The number of cells that are present in, say, for example, the cerebral cortex is reduced. The types of cells and their ability to differentiate is also reduced. And most profoundly is their synapses. So this is an image of a neuron, a single neuron with 20,000 synapses, and then that EM micrograph is what one synapse looks like. This is where all information coding in your brain takes place. And so these synapses tend to be abnormal in shape and size and function, and they're weak. And so the question is why is that so? The story that I'd like to tell you is actually one that's really a brainchild of a student in my laboratory, Fabian Fernandez, who was addressing this problem and looking at why the brain was not functioning. And he surveyed a lot of literature and tried to come up with a unifying concept that wasn't oriented simply about what gene was the memory gene that was disrupted. Rather, how do those genes together give rise to a change in phenotype, and whether you could address that through a pharmacological approach. And his interest then was focused on this picture of the synapse in the middle. He arrived at a very simple but profound conclusion, and that is that the problem did not lie in the ability of their excitatory synapses to encode information, but rather in the inhibitory nervous system being too strong. And this graph is shown where it gives you an idea of the functional output of your nervous system, in which is balanced by excitation and inhibition. If we have too much excitation, we have epilepsy. If we have too much inhibition, we have coma. And in Down syndrome, his prediction is that you're off-center. You're not too far away, but it's almost like you're on alcohol or on a benzo. And of course, if you're on a benzo, Valium, you don't learn very well. So that was his idea. And so he went about to try to test this concept. To do that, we took advantage of a mouse line that was been developed that actually is trisomy for over 50% of the genes that are trisomy in the human condition. And with this animal, he then designed a behavioral test that was mirrored by deficits in children with Down syndrome, something called object recognition. And it's a simple learning test, and I give a child two objects, and then I take one away, and I give them a new one. And if they recognize that they see a new one, they'll spend their time with the new object. And so this task is something that we could adapt to rodents. Rodents use this because they like novelty, they seek novelty, they need that novelty to find food and where they can find that food. If they go to the same place where they've been before, they're not going to be able to eat very much, right? So in this object recognition task, we would actually do it 1 day apart or 1 hour apart. You give them two objects today, you wait some time, and then the next day you give them one of the objects and then a new object. And if you do this in the gray bar, the light gray bar, for a normal mouse, they show a very high index of attention to novelty. Whereas the Down syndrome mice show no attention to novelty, and that number is very close to 0. So they'll spend equal time with the old and new object. However, if you now give, so this is now his experiment. So his experiment was, can I give low doses of different GABA receptor antagonists? So GABA receptors are important for inhibitory tone, and there are many of these drugs available. And so he looked at some of them, and he tested them by giving them once a day for 3 weeks, and then doing this novelty test. And when he did this, shown in the red bars, is a post-treatment assessment of whether learning has improved. And what you can see is that the height of both bars under this one drug is identical to the wild type. What this tells you is that the Down syndrome animals, after this treatment, could learn as well as their wild type counterpart. Even more importantly, on the right-hand side, is that after 2 months post-treatment, that learning was working as well as if the mice never had this problem. And what we think happened is that there was a neuroadaptive change that took place. When we were reducing inhibitory tone a little bit at a time, then eventually the system started to work again, and the systems are turning on and off normal. But before, it was too high. So this is actually a fantastic finding. It's a really cool finding, and it was an intellectually changing finding for the community, because up until this time, nobody thought you could change the outcome of having trisomy. So this was, for the community, has been game-changing. It happened in 2007, and people have thought really different about this ever since. At this time, we're like, well, this is cool. What are we going to do with this? How do we develop it? And fortunately, as Daria just mentioned, I was introduced to the SPARK program, and she's already told you about this, but what it really did was help us de-risk our program. And some of the things that it did are outlined here. It helped us to do preclinical assessment, to add value, introduced us to lawyers and venture capitalists, and eventually I used all this information. I went out to Roche, I went to all these places and tried to get people to help me license or use it, and I couldn't. And in the end, I founded my own company called Balanced Therapeutics. And over the last 3 years, we have been working to de-risk this project further. We now have completed our phase 2 clinical trial, which is outlined here. It took place in Australia, and we will be able to unblind that at the end of the summer. So I'm really looking forward to that outcome. What I really liked about this whole de-risking project is that whereas before Roche would, and Ciba-Geigy and all these other major companies who would not invest in our program have now recognized the value in what we're doing, and now Roche has its own clinical trial with its best compounds to address this problem. So my message to you is that even these complex disorders like Down syndrome can in fact be addressed if you really think about how the system is being affected. Now, the drugs that we're going to use or plan to use will not fix all of their problems. And I'm actually hoping for even a 10 or 15% improvement, because the quality of life would be huge if that was really the case. And so with that, I will just thank the many people who have helped me, in particular Fabian and Craig Heller, Dan Wetmore, and Lyndon Lin, my CEO of my company, and a lot of support. Thank you very much.
Thank you, Craig. That was great.